Okay, in the previous video we talked about thick, thin, and elastic myofilaments. And the confusing thing is now we're just going to call them thick filaments or thin filaments, but they are myofilaments. And so let's just backtrack for just a second. So the thick ones are made of myosin, and they are a bunch of myosins all wrapped together, and they have these weird little heads, kind of like golf clubs. And the thin myofilaments are actin and tropomyosin and troponin, but mostly we're going to focus on the actin in the thin filaments. And so when we put them together, we see this weird shape, and this is where we're going to start talking about what a sarcomere is, and we're going to start talking about those striations a little bit more. So the thick filaments and the thin filaments have this pattern, and they overlap kind of. Well, I shouldn't say overlap. They kind of line up like this, so it alternates thin, thick, as you can see on both sides. And then there is a bare zone where there's no thin filaments, it's just thick. And so this is showing a sarcomere, or part of one, not the whole thing. And so you can see really the size of the thick filaments compared to the thin. And so that bare zone that we see is going to be the part of the striation that you're able to see under the microscope. And I think we're going to get to that after we talk about these proteins. And really this week we're going to talk about everything, but we're not going to really say a whole lot about what it does. And then next week we're going to get into what a contraction looks like. But it's going to be important that you memorize all of these parts to be able to keep up next week with what is happening during a contraction. So we know all of these are proteins, but when we talk about the contraction, the myosin and the actin are actually going to be the proteins that are doing the work. But then we also include tropomyosin and troponin in the conversation because they're what we call regulatory proteins. They're basically a switch that determines when the fiber can contract and when it can't. So if you go back and we looked at tropomyosin kind of covers the areas, or the active sites, or covers the actin from where the myosin can attach. And so that coverage basically keeps the contraction from occurring. So the contraction is activated by the release of calcium into the sarcoplasm, and the calcium has to be able to bind to the troponin. If it cannot, then the contraction cannot happen. The troponin changes shape and moves the tropomyosin off of the active sites on the actin. So technically during the contraction we're going to be talking about the myosin and the actin and how they touch each other, grab onto each other, but that wouldn't happen if the tropomyosin and the troponin really hadn't moved out of the way. Specifically, the tropomyosin moving out of the way. The troponin being activated by the calcium is what causes that tropomyosin to move. There are a bunch of other accessory proteins that are associated with these filaments. And so I'm going to focus on just dystrophin because it is the most clinically important. But these proteins basically are just helping. So they either anchor the myofilaments, they can regulate the length, help them line up correctly, but they're not the kind of the main stars of the show of contraction. But we'll talk about dystrophin for a little bit. It is linked to the actin in outermost myofilaments, to the transmembrane proteins, and eventually to the fibrous endomysium. So it basically holds the outer myofilaments to the endomysium, and it transfers forces of the muscle contraction to the connective tissue around the muscle cell. So it is important as far as that, forcing or transferring the forces of the contraction around of the muscle to the connective tissue. And so when the dystrophin is altered, if it has genetic defects, then that's where we see the disease muscular dystrophy. And those muscles are working for a while, but then depending on the type of muscular dystrophy, then they can weaken. So let's talk about those striations. So we see those stripes when we look at the microscope view of some of the muscle tissues, specifically skeletal muscle and cardiac muscle. Some muscles are striated and some aren't. But the arrangement of those proteins is what's different And that's why we see some striations in some muscles and not in other muscles. It's the arrangement of those proteins that makes the striations. So here's a whole sarcomere, and we can see those thick and thin filaments that we saw earlier, but now we can see a little bit better of an arrangement of those. And so the myosin and the actin are the proteins that are going to contract. They are occurring in all cells. They help with the cellular motility, mitosis, and then the movement or the transport of intracellular material. In skeleton and cardiac muscles, they are organized in a very precise way. So they have what's called an A band. In this picture, you're probably going to have to look in your lab manual because this picture is going to be super tiny. And so the A band is the dark, and that is the thick filaments. Specifically, where the thick and thin filaments overlap is especially dark. But really the A band is the whole width of the thick filament. That little, I don't know what it called on the previous slide, a bare zone I think is what it called, is the H band. It's the middle of the A band where there's no thin filaments overlapping, just thick. And then there's an M line, which is in the middle of the H band. Now we're going to be focused on A band, I band, and Z disc. So the M and the H are kind of extra. And so the A band is just the dark, which is the thick filaments. The I band is the lighter band. And it is where there is only thin filaments. There are no thick filaments. So I can always remember the A is for dark because there's an A in the word dark. And I is for light because there's the letter I in the word light. And then the Z disc kind of bisects the I band. It's right in the middle of the I band. And it provides anchor for those thin filaments and the elastic filaments to connect to. So when we use the word sarcomere, which is going to become really important when we talk about the contraction, the sarcomere is the length or the segment of the myofibril from one Z disc to the next. So it basically includes half of the I band, a whole A band, and then another half of an I band. And that is what we'll be looking at when we look at contraction. There it is a little bit closer. And so this is, you can see the A band is the thick, and then the I band is where there's just thin. And we're going to compare this with what's coming up on the next slide. And so this is it zoomed in a little bit. I don't really feel like, there we go. That's a connect. That's the actual microscope version. So let's go back for just a second. So there's our thick and thin filaments. And then here they are again in a real visual. So again, this is zoomed in a lot. You're not going to be able to see this with your microscope, but you should be able to see those striations, but you're not going to be able to get this close. But this is what a sarcomere would look like with a microscope. And so that A band is the dark, which is the thick filaments, and then the I band are just the thin filaments. So the sarcomere is the functional contractile unit of the muscle fiber. So that when we talk about contraction, we're going to be looking at one sarcomere. And the muscle shortens because individual sarcomeres shorten. They pull those Z discs closer to each other. So see those Z discs, they're two thin little lines on this picture. When the muscle contracts, those Z discs are going to become closer to each other. Basically, this bare zone is going to become unbare anymore. This is how the contraction is going to work. This bare zone and those thin filaments are going to move towards each other, pulling those Z discs close to each other. But again, we're going to save all the contraction for next week. So here's a microscopy of skeletal muscle, and he's added some labels for you, but I don't think they're great. I think they're really hard to see. Usually when you're looking at a slide, whatever color it's stained, whatever that color is, the stained lines are going to be the A band, and the white line or the negative color is going to be the I band, and then you should see the nuclei throughout the skeletal muscle. So sarcomere, again, is from Z disc to Z disc, and the muscles shorten, the cells shorten because their individual sarcomeres shorten. So the Z discs are pulled closer together as thick and thin filaments slide past each other. The thick and the thin filaments do not change length. That's a big misconception. They do not change length during shortening. Only the amount of overlap is what's changing. So the proteins are going to link together and pull themselves, and that is what allows this shortening to happen, or contraction. Okay, next week we're going to have the nerve-muscle relationship. And like I said before, it feels like next week has got a lot going on in it. I'm going to stick to the schedule, but I'm going to open up all those videos earlier, probably towards the end of this week, so that you can move ahead. But just these first 3 videos for this week, or just these 3 videos for this week, is what you need to cover for this week's quiz.